Hello, my name is Mark Feeley. As of about a year and a half ago, I was the Associate Director for the Bureau of Chemical Safety in the Health Products and branch of Health Canada. Our Bureau is primarily responsible for the risk assessment, risk management, and approval of chemicals in food for Canada. Subsequent to that, I have worked with the WHO-FAO Joint Expert Committee on Food Additives and assessing both food additives and contaminants. And during my time with Health Canada, I was also for the past 5 years, the head of the Canadian delegation to the Codex Committee on Contaminants in Food. So the purpose for today's brief presentation is to go over the safety assessment that has been conducted for the veterinary drug Zilpaterol. A number of assessments have been conducted by international organizations. Primarily, the Joint Expert Committee on Food Additives have reviewed Zilpaterol twice. Prior to the JECFA assessment, Zilpaterol was reviewed and approved for use in food-bearing animals by the Food and Drug Administration of the US, and also by the Veterinary Drug Directorate of Health Canada. So what we will now do is provide a brief overview of the information and the basis for the safety assessment for Zilpaterol. So Zilpaterol, which goes by the trade name Zalmax, and it's produced, the major manufacturer is Merck, is to increase weight gain, improve feed efficiency, and increase carcass leanness or decrease the fat content of cattle. And these are cattle that are typically held for the last 3 to 4 weeks in a feedlot under controlled conditions prior to being shipped off to an abattoir. So the Zilpaterol is not approved for use in lactating dairy cattle. It's it's primarily for cattle that have been selected out for meat production. So the main issue, Zilpaterol, it has 2 stereoisomers. They're the figures there on the right-hand side of the slide. It's classified as a beta-2 adrenergic agonist, so therefore it binds to beta-2 adrenergic receptors. So there's a number of receptors, and they occur in a large number of cells in the body, and primarily they work on the sympathetic nervous system and cause smooth muscle relaxation. So as most beta-2 adrenergic agonists or receptor binding agents, they cause relaxation of bronchial passages, vasodilation in muscles and liver, relaxation of uterine muscles, and release of insulin. So not Zilpaterol directly, but there are other structurally equivalent beta-2 adrenergic agonists that are used in humans primarily to treat asthma and pulmonary diseases. So the issue of being able to bind to beta-2 adrenergic receptors is very important because in terms of a safety assessment, these receptors are very well conserved from a phylogenetic perspective. So the protein structure of the receptor and the binding sites have very high sequence homology across all mammals. So therefore, it's anticipated that a drug such as Zilpaterol, which is active in cattle, would also be active in humans. So as we mentioned at the beginning, Zilpaterol and its Zilpaterol hydrochloride has been assessed by the Joint Expert Committee on Food Additives both in 2014 and 2015. And as usual with veterinary drugs, these assessments are done at the request of the Codex Committee for Residues of Veterinary Drugs in Food, both the CCR-VDF 21 and CCR-VDF 22 meetings. So what you might think of Zilpaterol as a beta-2 adrenergic agonist or receptor binding, it's similar to the response that you would get if an animal was exposed to something like adrenaline or epinephrine. So it binds, it has a very short-term action, and it tends to be cleared relatively quickly in terms of hours. So in the first meeting that JECFA held at the request of the Codex, they established an ADI of 0.04 microgram per kilogram body weight per day. So as typical for JECFA meetings involving vet drugs, part of the evaluation considers residues in food-bearing animals for which the use of the vet drug is being requested. In this case, it's cattle. So at the time of the 78th JECFA, there was no question about the safety, and the committee felt comfortable in developing a health-based guidance value, such as an ADI. However, they had questions remaining from the depletion study and the residue information that the petitioner had submitted. So because a health-based guidance value as an ADI has been established, and it's evident in the JECFA evaluation from the 78th meeting, there was a full toxicological data package submitted in support of Zilpaterol. So these data packages, which are consistent with the approval or the requested approval of any vet drug, include pharmacokinetic studies. They also include short and long-term toxicology studies. They include cancer bioassays, so studies directly or specifically addressing the potential for cancer. They look at developmental or teratogenic effects in animals. They look at reproductive and fertility toxicity. And very importantly, they also look at the potential for this drug or chemical to cause any genotoxicity. And these are a combination of both in vitro and in vivo assays. So after reviewing all that information, JECFA had decided that there was a series of studies that were done on human volunteers where they were exposed to progressively higher doses of Zilpaterol in a beverage. And they indicated that the lowest dose causing an adverse effect in these human volunteer studies was 0.76 microgram per kilogram body weight. It's a single dose or an acute exposure. And the effect was described as neurological, which is tremors, which is a standard effect associated with beta-2 adrenergic agonists. So I think important to realize is that even at this low AAL of 0.76 microgram per kilogram body weight, the effects were described as transitory and very slight. So progressively higher doses would increase the magnitude and duration of the tremors, but at this dose of less than a microgram per kilogram body weight, the effects were very slight. So JECFA decided to use a 20-fold uncertainty factor, and the breakdown of that was simply a standard 10-fold uncertainty factor, which is used for human uncertainty, so human inter-individual variability. And they added an additional 2-fold uncertainty factor, which is standard practice when the health-based guidance value was based on a low L and not a no L. So, so the ADI was established at the 78th meeting. Subsequently, JECFA received the additional residue data for the 81st meeting, and at that time were able to recommend maximum residue limits for the requested tissues for this veterinary drug. There's a slight difference in description, whereas the health-based guidance value initially established was considered to be an acceptable daily intake, so denoting lifetime exposure. JECFA considered it was consistent with their practice to also set an acute oral reference dose for this compound, so an ARFD. And typically these are meant to cover exposures over a 24-hour period, which is consistent with this type of chemical, considering it has a very short half-life in humans. So when you look at Zilpaterol following administration to cattle, what you find is that the main organ that contains residues is liver, next kidney, and both liver and kidney are significantly greater than muscle and fat. In fact, after a 48-hour withdrawal period, which is consistent with practices using veterinary drugs, residues could only be detected in liver and kidney. So there's a specific process that JECFA uses, and it's described in a document that's available online. It's the Joint FAO Expert Meeting on Dietary Exposure Assessment Methodologies for Residues of Veterinary Drugs. So in this, there's a description of the process on how to conduct global acute exposure assessments. So this is the GEADE, so global estimated acute dietary exposures, which basically relies on taking a conservative approach of a 95th percentile of a recommended or recorded intake of a particular tissue. That is then combined with the residue seen, and once again, this is using a 95th percentile, seen at a 72-hour withdrawal period, which were available and considered to be consistent with the reported use of Zilpaterol. So typical for veterinary drugs, there's an application to the food-bearing animal, then there's a time period where the effects take place, and then afterwards, prior to the animal being sacrificed, there is a withdrawal period which has to be adhered to and is part of the good veterinary practice use of any veterinary drug. So initially, when JECFA looked at these 72-hour residues, they found the following amounts in kidney, liver, and muscle. When they applied the global estimated acute dietary exposure amounts using a 60 kilogram body weight for adults and a 15 kilogram body weight for children, they found that the intake for children would exceed the acceptable daily intake. So the intake for adults was right at the ADI, or the acute oral reference dose, but it exceeded the reference dose in children. So subsequent to that, JECFA then reviewed further the depletion studies that have been submitted and selected the 95th percentile residues at 77 hours. So these are, it sort of denotes how quickly this drug is being cleared from animals. At the time of 77 hours, the residues had been reduced to the 3.3 parts per billion, 3.5, and 0.5 parts per billion in kidney, liver, and muscle. So when these values were plugged into the global estimated acute dietary exposures for adults and children, for the general population, it resulted in the 80% of the acute oral reference dose. And for children, it was 94% of the acute oral reference dose. It was also noted that, as reported in the depletion studies and the test studies on efficacy for Zilpaterol, that the 77 hours was consistent with currently approved withdrawal times. So this simply provides a bit more background on exactly what JECFA considered. And as I mentioned previously, there was pharmacokinetic data that was reviewed, and it showed that Zilpaterol has a very high bioavailability, so it's absorbed into blood almost completely from an oral exposure. And the maximum concentration in blood, the Cmax, is reached within about an hour. And more importantly, similar pharmacokinetic profiles are seen with acute and repeat doses. And that is consistent with chemicals that have relatively short half-lives. And in fact, the half-life in humans for Zilpaterol has been estimated to be only 3 to 4 hours. So therefore, if someone takes a dose of Zilpaterol, within approximately a day, it will be virtually absent. So a variety of studies were conducted as part of the data package that was submitted to JECFA for evaluation. And these are basically some of the effects that were noted. And it's not surprising, considering the phylogenetic similarity of beta-2 adrenergic receptors among mammals, that these NOELs and LOELs are relatively consistent. So you see in the experimental animals, they also produce effects that are consistent with, say, effects associated with administration of something like adrenaline or epinephrine, where you have increase in heart rate or decreases in blood pressure. And these are all occurring at relatively low doses, consistent with the binding of Zilpaterol to beta-2 adrenergic receptors. So always around 10 to 50 microgram per kilogram body weight per day. More importantly, when you look at the developmental toxicity and the reproductive toxicity and the potential for genetic toxicity, all these come out are either negative or come out at significantly higher doses than the doses that have been associated with typical beta adrenergic receptor responses. There was a series of studies that were available for review by JECFA, and these were done in human volunteers. And essentially what they reported was that they looked at a variety of cardiovascular effects, and this is both diastolic and systolic blood pressure, stroke volume, or the maximum amount of blood being pumped out by the heart rate, bronchodilation, tremor, etc. And when taking all of the human studies into consideration, the lowest dose that seemed to be producing an effect was, actually was no effect, was the 0.25 milligram per person. And in these experiments, they increased the dosage that were given to the volunteers all the way up to 2 milligrams. So the low AAL for increased heart rate, where you have a minor increase in heart rate of about 6 beats per minute, was seen at this dose, so 3.6 microgram per kilogram body weight. And typical of receptor binding characteristics, when you increase the dose, you increase the response. So when human volunteers were given a dose up to 2 milligram per person, you would see increased heart rate up to 44 beats per minute. So as these drugs are used to treat, or structurally similar drugs are used in human medicine to treat people with asthma to increase bronchodilation, there was also a series of volunteers with asthma that were also tested. And what was found in these studies is that the dose required to elicit an effect was lower than normal human volunteers that did not have asthma. In fact, the lowest dose, which produced no bronchodilation in asthmatics, was 0.76 microgram per kilogram body weight per day. So this same dose produced very mild transient tremors in the humans. And once again, the effects seen in the human volunteer studies were all consistent related to beta-2 adrenergic receptor binding. So JECFA decided to take the dose in asthmatics that produced the bronchodilation and the tremors, and in this case, considered the tremors to be the more significant effect on which to base its evaluation. So as we previously indicated, this dose was divided by an uncertainty factor of 20 to arrive at the acceptable daily intake or acute oral reference dose of 0.04 microgram per kilogram body weight per day. So Merck is the US company, and they initially petitioned FDA to approve Zilpaterol. The exact same data package was presented to FDA as JECFA had for evaluation. FDA decided to use the most sensitive endpoint as bronchodilation. So essentially what they did, this was the dose that FDA in conversion of the body weight to dose decided was closer to 0.83 microgram per kilogram body weight. And JECFA was more conservative. They selected this as, because the dose was done on a milligram body weight basis. So they decided that the dose that JECFA considered to be 0.76 was closer to actually 0.83. And which is consistent with certain regulatory agencies. They select the effect where you can clearly define a no observed effect level. So in this case, FDA used the no AEL for bronchodilation of 0.83. They used an uncertainty factor of only 10. So FDA is a little less conservative than JECFA in the fact that JECFA considered the tremors to be a more sensitive endpoint, and they increased the uncertainty factor. But in general, both organizations looked at the exact same studies, and it's not unusual to come to a slightly different conclusion. Specifically, JECFA tends to be more on the conservative side. So their health-based guidance value is about twofold lower than FDA's. And finally, I think the last slide, EFSA, I should probably, basically at the request of the European Commission, after the latest JECFA assessment, was asked to revisit Zilpaterol. And they did this knowing that there's a general prohibition for the use of antimicrobials and growth promoters in the European Union. And this is a standard, especially for growth promoters. And in that, they include the beta-2 adrenergic receptor binders. They consider this mainly to be an issue involving both human health and animal health. So EFSA, in their review in 2016, basically reiterated what acute reference dose that JECFA had set and the MRLs that they had recommended. And, excuse me, basically indicated that the approach followed by JECFA in setting MRLs for Zilpaterol appear to be scientifically robust, which is, previously they had referred to it as comprehensible, which is sort of a term that's really not describing the nature of the scientific assessment. So scientifically robust, I think, is quite a bit more straightforward in terms of deciding that the process used by JECFA was consistent with international guidelines of safety assessment of veterinary drugs. So the only thing that, there's two things that EFSA mentioned was that JECFA used a low AAL, and EFSA may have conducted dose response modeling for those same studies instead of just selecting a point of departure, which was a single dose. And they didn't complete their evaluation and actually indicate how that might change the point of departure that was used by JECFA in establishing the health-based guidance value. But it's more consistent now in toxicological evaluation to do a mathematical dose response modeling if the data is amenable to that. They also noted that the withdrawal period that JECFA selected should be more than 3 days, which basically means it should be more than 72 hours, which is consistent with the 77-hour withdrawal period that JECFA indicated was consistent with good veterinary practices with Zilpaterol. And finally, JECFA indicated that the available literature indicated that there may be adverse effects on animals for Zilpaterol. So potential for increased mortality, heart rate, respiration rate, and agonistic behavior, so aggressive behavior in cattle. However, they said that the available studies couldn't directly link Zilpaterol administration to these effects. Other organizations such as FDA and Health Canada also rely on a safety assessment of experimental animals and the animals that are selected for potential use of veterinary drugs. So there has to be minimal to no adverse effects in animals associated with the use in order to obtain approval. So, and there have been other examples, especially in Health Canada, where vet drugs have not been approved simply because the adverse effects in the livestock were considered to be too great. So there's no human health indications, but the animal health was questioned. Merck, the manufacturer of Zilpaterol, I understand, is currently conducting a more detailed evaluation, taking into consideration all the reported literature to exactly indicate what, if any, adverse effects have been associated with the use of Zilpaterol to date. And we might add that, not barring any further discussion by Codex and JECFA on this issue, that Zilpaterol is currently used in a number of countries worldwide and has approval for use primarily in cattle, although manufacturers are looking at increasing the number of animal species that Zilpaterol could potentially be of benefit for. And that, I believe, is the end. So I'd like to thank you for your time in reviewing this presentation. Hopefully it's answered any questions you might have had regarding the safety assessment for Zilpaterol that's been conducted, in particular by the Joint Expert Committee on Food Additives. And we'll provide a bit more background details on how the MRLs were developed, which are currently under discussion with the Codex committees. Thank you.